One-dimensional line. I'm saying find a problem E, right, left, and jump left, right. Did you guys try homework? Problem number 2. So that problem you see that you can change the probability of jumping right and left. Now there could be 2 constraints you can impose. One possibility is the sum of PR plus PL must add up to 1, or PR that means you might have a situation where you have 4 L and then you have a small probability of continuity. So you can go left and right to probability 1.4, but there could be a probability which you just stay there. And we discussed in last week's lecture how to simulate this process, this type of processes, and we're doing more problem 2 on this. Now the next thing we are going to do is to show you how you can calculate statistical such as average or variance. Stochastic process. Now I'm going to show you that a little bit later today. So what sort of biological or biomedical problems or questions kind of motivate such Monte Carlo simulations? This is a very simplest scenario of Monte Carlo, like a one-dimensional random walk, and in real life you don't have such simple things, but you can approximate even more quite complicated biological processes using this type of random walk diffusion and reaction. And I can show you what type of things you can do. And there you'll see there are 2 things you can extract from such simulations. One, you can start a simulation and watch different molecules move and cluster and react, those kind of things. So you can kind of dynamically follow different molecules. So that's one thing. And the second thing you can do, you can calculate statistical quantities, and that can also give you meaningful information about a biological system. And it will be clearer once I give you very concrete examples. But before I show you real examples where these kind of Monte Carlo simulations are used, let me tell you a little bit about these statistical quantities you can calculate using Monte Carlo simulation. And that will be the next exercise we are going to discuss in this week's computer lab, and also to be part homework 3. Homework 3 will see similar problems. You need to calculate statistical quantities. And from 105, you are aware of this kind of quantities, correct? So if I give you this process, suppose I am crossing one point, and I define the random variable. I think I did similar things in my last week's computer lab. So x equals 1 when a, and x equals minus 1 for k. You know probability of a is f probability of a. This is going to be a bit kind of refreshing your memory from 105. So how do you calculate x average from this process? The way you do it is write sum x dx. So x is the value random variable takes, and px is the probability. Okay. So in this case, this random variable takes two values, 1 and -1, and you have two probabilities. Both are high. Okay. So you can write this sum as 1 half minus 1 Are you with me? So this is expanding this x dx. So x times px, and then x into 1 times px. That makes sense, correct? For tossing a coin, it makes complete sense. Because even if you, suppose you are tossing a coin, initially someone is getting some heads, it could be by chance. But if you toss the coin a million times, you know that on the average you get equal number of heads and tails. And that's what is reflected here in the average. But not the variance. The variance is not going to be 0. So if you calculate a square, square average does not vanish, and you can calculate the variance. What is variance in terms of x squared and x? Do you know? Any of you here? Correct. But in terms of x squared average and x average, how do I write the variance? So I already calculate x squared average, and I have x average.
X squared average minus x average?
Minus x average squared, x average squared. X squared average. So you see that variance is finite, even if the average is 0. And you can do that for all sorts of probability distributions. I give you x random variable. Which takes value of x, that's defined by random variable. So random variable is defined by two things. One is the value it takes and the probability for that value. Okay. So if you are defining the probability distribution P of x, then you know how to calculate x average and x square average. For discrete distribution in some xpx, for continuous distributions, integral x. And you must have seen such things in 105. Yes? Now, what does these quantities mean, especially the variance and the average? And think of random walk. What variance, average, and variance will tell you if you know those quantities.
Is it like the average distance will travel like 0?
Correct, but so the average will mean for a random walk that if you plot, so you have done these simulations in computer lab, so individual trajectories are everywhere, like they are all over. You plot x versus time. Now the average is at 0 for random walk. That's the average. So average is a measure of kind of what it, from its name it's clear, it's the average of all trajectories. Whereas variance measure the fluctuation, how much you fluctuate from average. Okay. You have this average at x equals 0, and the variance means how much you are fluctuating. Individual trajectories are going to fluctuate around the average. Makes sense? So that's one thing you should keep in mind. That average, by its name, is kind of the average behavior of a system. And the variance describes how much you fluctuate around that average behavior. Okay. If I describe a motion of this chalk under gravity, then the variance is 0 for that motion, because it's deterministic motion. Okay? So the average determines everything. And the other extreme is random walk, where the variance determines everything, because the average is not representative of these large fluctuations. So the variance gives you the real measure of the system. So a stochastic system, most important, if it's very stochastic, then the variance dominates, and that's very important. But also the average behavior you can find out is fluctuating around which mean or average value. So both are important for the stochastic process. And even without calculating, in this case, you have probability of x is and the average is here. Average is at x equals 0. This is average. And you see that this could be a measure of your fluctuation in the system. So here is your average, and it can take either +1 or -1. So that means this is your spread in the system. And that is, so spread is of the order of 2. That is fine. And that kind of gives you an idea of your variance. Your variance is also 1. Gives you some idea. Or you can even think of delta not from the between two extremes. This is more like range. So range between two extremes is 2, but if you consider the variance from average to here, this delta is 1. Or in the other direction. So on the average, you can go plus or minus 1. So that kind of gives you an idea of the variance. And that's what you get. Now, how do you calculate average and variance for a random walk? Calculate means analytically. You can of course run simulations, and I'm going to show you in this week's lecture how to calculate this quantity from the simulation. Okay. But what I'm going to show you now is how do you calculate things analytically. And the question you might ask, like, why do you care? Why do you want to calculate things analytically? When you have a computer, you simulate the process and you just calculate the average or variance. So, any thought on this? Like, why do you care to calculate things analytically for simple systems?
Is faster?
Could be, depending on your proficiency in doing analytical calculation versus running computer simulation. But
to be able to do just like back and matching things to just estimate it.
now, one thing is when you are doing computer simulation, you don't really. It's like so you guys did differential equations. Yes. How about numerical solutions of differential equations? Like Euler's scheme, did you do that? Now, if I give you this equation, dy dx dx, you can readily write the solution. But if I give you this differential equation, that you cannot see the solution right away. And it's possible that this equation may not be exactly solvable, like analytically solvable. Then what you do, and we're going to discuss those things in detail when we go over here. So there are a lot of complicated nonlinear differential equations for which you cannot do analytical solution like this. So what you do numerical solution, right? But to know whether your numerical solution scheme works, what you do, you first try your numerical solution with known cases, meaning for which you know analytical solution, so that you can compare. Okay, so you do numerical solution for simple cases, and you compare and check your result with the simple solution. And if it matches well, then you gain some confidence that your numerical scheme is right, and then you go and start solving more complicated equations for which you don't know the analytical solution. Makes sense? And the same idea here. So if you know analytical solution, then you have a lot more confidence and understanding of the underlying problem. Yeah. So when you're applying this Monte Carlo for more complicated biological systems, for which you will not be able to solve analytically, or not easily. Okay? But you already checked your results, your simulation results, in simple cases. So it's a much more rigorous approach to first do simple problems both ways, both simulation and analytical, and check your answer. And if you think that it's working well, then you go and develop more complex Monte Carlo simulations. So that's the idea. And let's say if we could calculate these quantities for very simple cases. Now, suppose this random walk x n steps and x steps toward now. Do you know how to write this probability? Xn you follow my question? So you are taking steps right and left with this probability, and suppose you were n steps, and out of n steps you write how many steps you took to the right. That's the average. You are correct. That's the average. On average, you are going to take n over 2 steps to the right. But what is the probability distribution that you take x steps to the right out of n trials? And it's like a success-failure type probability distribution. So either you toss head like a success, or you toss tail like a failure. And what type of distribution are you familiar with? Binomial, correct. So it's a binomial distribution. It has two outcomes. Head, which you can call like success, and then tail. And typically when you have this kind of two outcome systems, it's a binomial distribution. And the probability of binomial distribution is P equals half and Q equals half, but you could in general write it arbitrary P and Q. So what's the probability of taking x steps to the right, or x success out of n attempts? Do you recall? It's n of cx p to the power x, c to the power n minus x. Does this look familiar? Did you see that in 105? Yes? I'm going to so NCX means n factorial, x factorial. Question? Do you follow? The back? Yes?
So Q to the n minus x.
Q to the power n minus x. This, can you calculate? Let me first do the average. Do you remember? But you already told. But in terms of P, how do you write it? N times P. Okay. N is the total number of attempts, and P is the probability of success. It makes sense to have this probability as NP. This I will ask you to go back and refresh your memory and check how you calculate it. But even if I don't do rigorous calculation, the average I can estimate from simple dimensional arguments. That means X is probability of success. So if P increases, X must increase. Do you follow? So if P also goes up. And if n increases, x average should also go up. Because if you have large number of trials, your chances of success will also increase. And if you have higher probability of success, your chances of success will increase, average, right? So that means this quantity has to depend on n and p. Now it could have been n to the power alpha and p to the power beta in the most general situation. But the simplest choice you can say is NP. So even without doing rigorous calculation, from dimensional argument, you should be able to guess the average in this case. Now, x square average in this case is I'm not going to show you the derivations of this. I gave you some motivation why x average is going to be NP, but you can calculate using this probability distribution. Starting from this, you should be able to do that, and that was already part of your 105. So I'm not going to repeat that exercise here. Okay. Now, the next thing I'm going to ask, so why your x average is not 0? You follow the question? My question is why x average is not 0, because if they are moving with equal probability left and right, why x average is not 0?
Because you're looking at number of steps taken in the back of your own displacement.
Correct. That's exactly right. So x is a random variable, which is the number of steps towards right. That is not going to be 0. That's not the displacement. So we need to define something as a displacement operator, which is, let's say, R. And how do I write R in terms of x and n? So what is R? R means net displacement towards right. Okay. So that means I need to do steps to right minus steps left. Are you okay with this? So you need to subtract steps to left from steps to right. That's the net movement, net displacement. Now steps to right is my old random variable x. And what is steps to left? What is that? That is n minus x. Okay? Because you have taken total n steps. If x is towards right, n minus x must be towards left. Total is n. So x to left, to right, that means n minus x to left. And now x minus n minus x is 2x minus n. average. Let's first calculate the average of R. X average minus N. And what is X average? That's NP from the top board. It's 2 NP minus N. Okay. Now if B is half, correct? So if you have half probability of moving left and right, then the average displacement is 0. But it's clear from here that if B is larger or less than half, then the average displacement is not 0. Then you have some drift either towards right or left. Okay. Now if I calculate R square is 2x minus n whole square, which is 4 square plus xn square. Square average is n square is a constant, so if I take the average, it stays n square. X square average, I calculated NPQ plus n square P square. Minus 4x average is NP. Check what happens for unbiased coin toss. What E equals half, I get 4n square square minus 4. Please check my calculation when I used numerical factor, but the answer makes sense to me. Could be off by factor of 2 or something. Seems correct. So two kind of major results we get from this exercise. One is the average displacement is 0. That's one. And 2 is average square displacement, or the average of displacement squared increases with n number of steps. And these results make much sense if you simulate random walk trajectories or think about random walk problems and try to correlate these two results, the statistical average. Anyone? Yes.
That should be harder. Or one.
Where? So the first one I already discussed in the computer lab classes. You have a random walk. And you are going right and left with equal probability. On average, you are not moving. Okay? So the displacement start from 0, and you are going left and right with probability you have, and you are measuring the distance r at step n, at time step n or trial n. You are measuring how far you moved from your origin. Now, because you are going left and right with probability half, on the average, we see that the trajectories don't move. The average, if you sum all the trajectories, you get 0. Okay. So that kind of correlates with this result. Now, how do you kind of make sense of this second r square increases or is proportional to n number of steps? Any thought on that? So the way you have to think about that is consider random walk trajectories. As you go further and further in time, the chances that you are going to drift away from this average increases. Does this make sense? If you are confined to small time steps, you really didn't go far from the origin because you have taken only finite steps. But if you are waiting long enough, there will be 1 or 2 trajectories for which you will drift farther apart, far away from your average, from your origin. Okay. If you wait long enough, not for all trajectories, but some. Either you will go far away in this direction or go far away in the negative direction. That means with time, there will be few trajectories which will drift far away from the origin, and that will give rise to this large variance here. So as n increases, the number of steps increases, your variance will also increase. It means the fluctuation in the system around the average, how much you will fluctuate around the average, that's going to increase linearly with time. And this is a very basic but very important result in random walk distribution. So if you think n like time, so your displacement squared average increases with time. And later when we discuss partial differential equations, I'll show you that even for diffusion equation, you see the same result. Okay? Because diffusion, you can think of nothing but a collection of random walks. So why do I say that? take a beaker filled with water and you drop some ink here, and it dissolves and diffuses, and after some time you get uniform distribution. Or if you place some gas molecules in the corner of a room, after some time the gas molecules are everywhere in the room, and that's because diffusion. And you can think of diffusion like collection of random walkers who starts here, but with time they are moving farther and farther away from the origin. So if you plot the probability distribution, at equals 0, everything is at 0. After some time, because they are moving back and forth, you'll see a spread. If you wait longer, you'll see even longer spread. Like that. This is 0, this is 1, this is 2. So as you go in time, you go further and further away from the origin, and your probability of finding the object is higher further away from the origin. So initially everything is concentrated here, and then with time things are spreading due to random walk. Okay? And the same idea is in diffusion. When I solve partial differential equations, I'll show you that that's the solution you'll get if you solve diffusion equation. So the y-axis is probability of x. Probability of finding the random walker at x or x a. So now we probably get some idea that random walk is or can be a model of diffusion, which is everywhere in biological systems. So you can use random walk to model the diffusion process. It could be on the cell surface in a bunch of receptor molecules, and they are going to diffuse on the cell surface randomly. And that diffusion process you can model using a Monte Carlo of random walk. Any questions? Let me know if this is clear to you, this result and this result, and why it makes sense that the average increases with n or the time step. These are very crucial things which we are going to use often. Is clear? Makes sense? Back? Now, if I get time, either later today or next week, I'm going to show you another approach of calculating the same thing. But the advantage of that is in a way closer to Monte Carlo simulation in split. Okay. That's called the master equation approach. If I get time, I'm at least going to give you some idea of how that is done. And now the question is, can you now calculate this from the simulation? So far what you have done, you have generated these trajectories. This you know how to simulate. Now using this information, can you calculate R average and R square average? Do you follow my question? So why don't you think about that? Using information of these trajectories, how do you calculate this on your computer, these two quantities? And then you'll be able to check whether you will get the same answer as we get from analytical calculation. Right? That's the idea. That you should be able to check your results both from simulation and from analytical arguments. You should get the same result. So why don't you think for 5 minutes about this, and we'll take a break after that.
I've had a lot of practice.
What if like bear looking thing without eyes?
So like,
A, very long
She pulls around wherever you want.
We're gonna learn all the secrets with
Monte Carlo promotion.
This is high.
No, I'm using the rocks.
Pigs
I'm shooting birds at pigs. I'm using the rocks to kill them. See the strategy?
Why don't you shoot birds and pigs?
Dude, I thought about that, but it's not working out so well. There's one more left and I have one bird.
so then this is,
Oh.
so then it's just the sum, right? So you know that up until this
Those are pigs.
it's
There's green pigs.
plus this, right? So
Look
basically,
at that. See that? Oh,
so adding zero,
oh,
zero plus and then it's one.
You're like 30 and you don't 35.
Based on
30%.
I thought it was so you haven't told me
That also sounds
no, no, you might need that. Kind of like, where are you going with this?
Library
Oh yeah. Well, I have to probably already you still get it?
So, instead of doing
that list. And then that. And then
yeah, it's probably not safe for work. It's like, it's really good drawing. Like, you'll know what I mean. Give me a problem. There's a movable one out. I've done it before. Two operators. Watch this. This is like an aerial assault. Watch. There's no way. Only if it's a certain kind of you gotta keep up. How am I supposed to get that there?
So I'm going to give you like stuff where people, and also my own research lab is using Monte Carlo simulation. So what you see here, are you familiar with immune cells? Like T cells, B cells? Do you know what they do? What do they do?
So you can't destroy foreign bodies and use the bodies.
So T cells and B cells are adaptive immune cells, and they can recognize foreign antigens, means virus proteins. Okay? Proteins derived from foreign pathogens. And what T cells do, they activate a series of critical mechanisms that activate other cells, which can in turn kill those virus infected cells. And B cells produce, do you know what B cells do? What do they produce? Antibodies. So B cells are known for producing antibodies in our body. So when you have a cold or flu, the antibodies are all secreted by B cells. And what is the first step of this immune reaction? The very first event, if something gets into our body, which is harmful, cold or flu virus. T cells and B cells trigger this immune response, they activate other cells, and B cells secrete antibodies. But what is the very first event of this immune response? The very first event is a recognition process, meaning what you see here, a B cell between two antigen-presenting cells. Okay. So these antigen-presenting cells, or APCs, they show the foreign proteins on their surface, Okay? And B cells or T cells, they recognize those surface proteins. Okay? So that's the first and probably one of the most important events in adaptive immunity, because this process controls the recognition. So first B cells needs to know that, okay, I find something foreign and I need to trigger immune response. And that's this step where the B cell is recognizing foreign proteins on the surface of antigen-presenting cells. Okay, so this is, you can think, one of the most important events in immune response. So this event will control whether you will have a productive immune response or not. And if you don't, often you have diseases. If you have less than optimal immune response, your disease will persist. If you have more than optimal, you'll suffer from autoimmune disorders, okay, or other disorders, or if it attacks your own self issues. So very important to kind of finely tune or finely balance this recognition and signaling. Now, what's important and interesting that imaging experiments showed that during this recognition, there is a nice clustering of receptor molecules. So what you see on the right panel, a bunch of imaging pictures from imaging experiments where you attach a green fluorescent tag to basal receptor, which recognizes the antigen. Okay? Or you attach to the antigen itself. Actually, in this image, the tag is attached to antigen. And red is another set of molecules called integrins, which hold these two cells together, which help these two cells stay in contact for a long time and also signal. Okay. Now, you can imagine that when two cells are in contact, there will be receptor ligand binding, and receptors will flow from this region to the region of central cell-cell junction. Whereas you imagine a hypothetical scenario where imagine cell surfaces or artificial bilayers are created in such a manner that you have kind of flat contact between two surfaces. Do you follow? In one case, you have two cell surfaces or two bilayers in contact, but it's flat cell contact. In the other case, you have a curved region. And if I tell you this, that in this case, when you have two flat surfaces, receptor molecules will bind with antigen everywhere. And if you image, you will not see any clustering. You will see uniform fluorescence everywhere. But if you image in this situation, then you will see more fluorescence at the junction, less if you go away from there. Any idea why, of the mechanism, why that's to be the case? Let me give you a little bit more. So imagine this receptor ligand bond as springs. Now what happens if you stretch a spring? It tries to come back to its equilibrium position. If you have a spring, you stretch it by S, it tries to go back to its natural length. And there is a force of negative kx that will drive the spring back to its natural position. And if I imagine this as springs, there will be kind of optimal length for which you have maximum binding. Now if you go away from this region, what is going to happen? You can still have binding, but what do you have to do? You need to stretch a lot as you go farther away. Now you should be able to tell me why I see clustering at the center. Is the lowest energy possible? Why is a spring, if you stretch it, it tries to go back, because that's the minimum energy. If I drop this chalk, it goes to the floor, because that's the minimum energy. The same idea here, that because of energy, it's going to go. But it's not only guided by energy, there is a finite temperature, and the mass of these molecules are not really large like this chalk. So they will also try to move around, diffuse due to entropy. Okay. So there will be competition between these two, but still the energetic terms will dominate, and at least in room temperature or physiological temperature, you'll see clustering of this set of molecules here. Whereas if you have two flat surfaces, you don't have any preferred height. So you can imagine is the natural length everywhere. So there is no reason why things should cluster in this location, not here. Okay? There is no apparent reason of clustering. Right? Okay. So that's first kind of step in our understanding of this pattern or this clustering. But there is even more interesting one, what makes it worth studying through complex simulations is if you imagine for curved cell-cell contact, you circle like and bonds towards the cell-cell junction. But now I'm imaging two sets of proteins. One is green fluorescent and the other tag is red fluorescent. Now everything should go towards the center. Why you have one set of molecules at the center and then another set kind of in a ring. So what happens here, you have molecules of type A at the center and then molecules of type B outside. That's the side view. Okay. What you see here is the top-down view. Okay. So you see there's a top-down view, as it is one set of molecules here, then the other set is in this region, in the ring, in this ring. So inside a circle of type A, which is green, and then there is a type B, which is in red. And if you look at sidewise, you will see like this. One set of molecules here, and the other sets are outside. Now I will ask how you separate those two types of molecules, because all will try to go to the center. How do you segregate them? So that's where Monte Carlo simulation can be very effective. So what we can do on a computer, we can simulate this process. What we do, we create a surface, just like you are doing one-dimensional chain. Place bunch of molecules, and then they diffuse. Exactly like the way you are simulating a random walk trajectory. They go left and right, but now it's not in 1D. Cell surface is 2D. So the simulations are done on a 2D surface. Okay. So on a 2D surface, there's a bunch of molecules. And then they diffuse both in X and Y. And then I have another surface where I place another set of molecules. And when molecule of type, say 1 and molecule of type 2 are in contact, they can form a complex paper type B. Okay? So they can bind also. So they diffuse and bind. Now what's the advantage here? Now you can change various parameters in isolation, meaning I can first try to see if I make A molecule smaller than B molecule, what happens? Do I get a segregation? Then I do another scenario where I keep the sizes same, but the affinity with which they bind, which is, you can think like a spring constant. So if you stretch a spring, constant A, so any spring, if you stretch, it's trying to go back to its minimum energy. But the spring with higher stiffness will move back faster. Okay. And the same idea here. There could be various properties of these bonds which you can change in a very controlled experiment. And that's difficult to do in biological experiments. Because when you are imaging this, you have everything. Like diffusion, there are molecules, and there could be size difference, there could be difference in affinity. Now, how do you not do size difference? How do you block size difference and only check if affinity difference is enough to create this pattern? That's difficult to do in biological experiments. But very easy to do on a computer because you can change the parameter sets in your simulation, just like you can change the ER or PL in your random walk simulation. You can change those size parameter or affinity parameter and see whether you get this pattern. Does this make sense? And then later you can also do more. You can think of, okay, so this pattern leads to optimal immune response. And in some situations, suppose you're trying to design a therapy for a disease, and you want to change this pattern so that the immune response is either increased or decreased. And you want to do a diffusion study. So you want to suppose create a medicine which changes the diffusion constant of one of these molecules, and you want to see what's the effect. And you can do it easily that on a computer. You can change one parameter like PR or PL and see how that changes B cell immune response, how that changes antibody generation. Okay. Of course, that's not easy, and such is like we are kind of working towards that goal, but you might be one day, if you can use computer, change one of the diffusion constant using a therapeutic agent and see its efficacy, see whether that's going to give you the right immune response you want from these cells. And here you see another set of experiments. So these experiments are done by other labs. I'm not showing our results. I'm giving you some idea, but once you do a little bit more complicated Monte Carlo simulations this week and next week, then I will show you more of our own results from Monte Carlo simulation. Once you have that background, it will be easier for you to follow. But this is again experimental results from other labs, where you see that this pattern is formed over 30 minutes, like it's stable over 30 minutes. So it forms around 3 to 5 minutes, and then it stays there for 30 minutes. Okay, so this is the dynamic time course of this pattern, this cluster. Get some idea, then let me tell you, we also on another project that we are working on. So this is a study of very basic and important cell biological process, cell death. Are you familiar with this? Can anyone tell me anything you know about this pathway, or what happens if it goes aberrant, like if there is something wrong with this signaling pathway, what happens? You know? Right. So it is linked to a lot of very serious diseases. So you, for example, in tumor cells, it's often thought as a disease of apoptosis signaling, because what happens in tumor cells, you don't have apoptosis. Apoptosis is somehow blocked. And And it could be mediated in normal cells, it could be mediated by this kind of agents like receptor ligand binding or stress signal or toxic agents secreted by killer cells, like immune cells. This is a kind of simplified picture of the apoptotic pathway, where on the left you see there are two pathways. One is the type 1 or extrinsic, and then there is type 2 or intrinsic. Okay. Now, intrinsic pathway or extrinsic pathway starts at caspase 8 and goes directly to activate caspase 3. Do you see that? It goes from caspase 8 to caspase 3 or 7 in one step. That's the type 1 pathway. And then the type 2 pathway is more complicated. It goes from caspase 8 to bead to BCL2, TBEAD, and then there is a picture of mitochondria over here. And mitochondria releases a molecule called cytochrome C, which which binds with HAPAF1, creates a complex called apoptosome, and that eventually leads to the same caspase activation. Now the question you ask, how apoptosis, something happens at the level of receptor ligand binding and you have some active caspase 8, does the signal goes through type 1 pathway or type 2 pathway? Do you follow the question? So you have like two channels, like two hands, and signal can go through either of them, and you want to understand how signaling is controlled through these two channels. Now, let me tell you some experimental information. The first modeling attempt that we did was kind of designed to answer one kind of very basic question, that in some system, apoptosis was found to be very slow. It could take 10 hours. And in some other system, it's very fast. It takes 10 minutes. And the mechanism, why it's very slow in some systems, in some like cell death of some type of cells, or under certain apoptotic stimulus it's very slow, whereas in some other cases it's very fast. So that's one of the kind of question we had in mind when we first developed this model. And then the question is how these two pathways differentially get activated. And that's kind of the power of systems biology. Previously, these pathways have been studied by thousands of people and thousands of times. And what they did very important things, but they were focusing on a very specific part of this pathway. So probably they are studying how apoptosome is formed, or how cytochrome C is released from mitochondria. Do you understand? It's like bits and pieces. They're focusing on a specific part of the pathway and trying to understand what's going on in that particular place of the network. Okay. Now, when you put everything together, how the whole pathway interact with hundreds of different molecules. So that's not trivial. And that's one of the very powerful kind of approach of systems biology, which can provide very important insight into processes. And especially for this one, I will show you why computational systems biology is also very effective. Okay. So I'll give you a few minutes to think about what you could do easily when you have such a complex network and two pathways using computers, like the way you are doing multiple simulations. So keep two things in mind. One is the experimental observation that you have very fast and very slow apoptosis in some sense. But more than that, you see this picture and think, and I have two channels, and I want to study how apoptosis is activated in channel 1 or type 1 versus type what I could do using a computer very easily, not very easily, but probably easier than what you could do in experiments, but study the behavior of type 1 and type 2 separately. First of all, do you get my point? Do you understand the question? Yes? Good. Something.
We were just talking about like if you were to program it, either just using one channel and change channels individually, or using some sort of probability to say what channel to do.
Yes, that's exactly right. So that, and how do you do that on a computer? That's not much different from changing what you are doing in homework set 2, changing here and here. Okay. So you have a random walk and you can easily change the parameters of going left versus right. And it's the same idea. When I simulate this whole process, at the very first step, I will have a bunch of signaling reactions and a bunch of probability constants. So I have this molecule, gas phase A, and then I have either this reaction, B going to PB, or gas phase G inactive going to gas phase G active, which is called by gas phase G star. So you can go to either type 1 or type 2. Now, how do I simulate this? You will see, you will get the idea, better idea later when I do more Monte Carlo simulations. But let me tell you what we have. We have one binary constants like P on 1, and then for type 2, I have another constant called P on 2. So these are like probability of going left and right in a random walk. Okay, so P1 on means how quickly I can attach gas phase 8 to gas phase 3. And P on 2 tells me how quickly I can attach gas phase 8 to B. Okay. So these two constants kind of determine how fast I react in channel 1 versus channel 2. Now in real experiments, it's very difficult to vary this probability because these are molecules which are designed and part of the pathway. you're not going to change their affinity or reaction constant. It's difficult, much more difficult. Whereas here, everything is in my hand. I can change these constants, and what we can do, as the best option, is I set this to 0. That means type 2 is completely blocked, and I study only type 1. And then I do P1 is completely 0 and open P12. Type 1 is completely blocked. I only study type 2. Okay. And that's very difficult to achieve in real experiments. How do you completely block? You can, but it's much more complicated. And you try to block with some agent, and that agent not only blocks this pathway, but it does 1,000 other things. So disturb the complete system. Whereas on computer, I do it in a very clean manner. Okay? I block this and study what's happening here. Block type 1, I study what's happening in type 2. And then I can vary these two constants and see how gradually the signal goes between type 1 and type 2. Okay. And from that study, we could deduce that for strong stimulus, the activation happens through the type 1 pathway, through this direct pathway. And it's fast. Okay? It takes few minutes. And when I block type 1 pathway, and I study only type 2, I see very slow activation. Okay? It takes hours. So that kind of solves two problems. One, I understand how these two channels work. Second, I get some insight why apoptosis is very fast in some systems and very slow in others. That means when you have strong stimulus and this pathway gets activated. So that's another thing we see. When you combine two, when both pathways are open, under strong stimulus I go this pathway, under weak stimulus I go that pathway, so for type 2. So I know that if I have a strong stimulus, and if I go through type 1, I will see fast activation. Or if I have weak stimulus, or somehow I don't go from caspase 8, somehow I activate directly somewhere in between. So basically, if I activate type 2, I will have slow epitopic. So two questions I can answer from this study, and you see the power where I can change this kinetic constant, like set 1 to 0 and study the like one of the pathways by setting the kinetic constant to 0. Now I can one result. So that's how the simulation is done. I create a lattice. It's 3D, but it's not much difficult, much more complicated than your 1D. So if you can simulate random walk in 1D, and this week I will give you problem set 3, you will see 2D random walk simulation, and also this week's computer lab, I will focus on 2D random walks. And then you can increase the dimension to 3D. So you have a box, and basically the same type of move. You go left and right, up, down, like that. And then you have a bunch of probabilities for reactions, with P on, P off, P catalysis. Okay? And then you follow the time course of this signaling molecules. And this is kind of the initial result. I'm not going to show you more than this. So what we see when you block type 2, type 1 is very fast. It takes about 2 minutes. And when you block type 1, type 2 is very slow. But there is another interesting thing, more than only slow and fast. Different colors in these two pictures represent different cells. Like you can imagine, each simulation is representing one single cell. So these are single cell type results. Yeah. So, and the solid black curve in both left and right panel, those are the averages. Just like in random walk. Bunch of trajectories, and then the average is x equals 0. the average. And these are different trajectories. Think these simulations like that. So each color is like individual random walk trajectories or individual cell signaling. And then the black curve is the average. Now, can you tell me a basic difference between these two sets of pictures? This like between left and right hand. Meaning in terms of single cell activations. Right, that's exactly the case. So, and if you are to, if I ask you to compare with the random walk, which one is similar to random walk? Two, right? So if type 1, as you are telling me, that you will see the different colors are kind of close to each other. And the black curve is sort of, it captures the basic behavior, right? It captures the basic behavior. But on the right, the black curve does not really represent each individual cells. That's more like random walk. You have some kind of average, but individual trajectories are everywhere. It's much more dominated by fluctuations. Okay? And the same thing here. You'll see one cell got activated by 1 hour, and some other cells got activated by say 10 hours. And now you could have, you could find that in the type 2 is similar to type 1. Like you have fluctuation, so all cells are getting activated slowly, but that's not the case. It's more like one cell got fully activated by 1 hour, then nothing happened to some cells, got activated at 2 hours, some cells nothing happened 4 hours, then activated, then some cells at 10 hours and suddenly got activated. So it's very different from the type 1 behavior. Right? Now, let me see if I can quickly start the discussion of the master equation. I'm going to come back and show you more results. And one problem I can tell you to think about, which we are studying right now with my experimental collaborator, that is in cancer cells, there are two things. One is apoptosis is blocked, so you don't see apoptotic here. That's one. And then a big challenge is to try to kill cancer cells using drugs. And there are a lot of drugs which in the market right now which target this pathway, this type 2 pathway. So a lot of cancer drugs will try to kill the cell by activating the type 2 pathway. And a major challenge is that the cancer has billions of cells, and you can kill only a fraction of those, not all cells. And that could lead to relapse with a more serious form. And think about this, like why it's difficult to kill cancer cells, and there is always a selective killing, meaning only a fraction of cells will get killed, not all. Okay, think about that, and we'll come back and discuss the answer, which we are thinking right now, kind of what is happening in cancer cells in a week or two. Some of the motivations of why we study things on computer using this kind of Monte Carlo stochastic approaches. And once you do some more complex simulations, we will get a better idea of how to approach these problems. There's a second approach, which I'm not going to spend too much time on, but which can also describes such stochastic systems. The approach based on master equation. And this equation is different from regular differential equation, like ordinary differential equation. This is different. And what master equation gives you, it is an equation in probability of x and t. Probability of finding molecule at x and so this is the probability that you find the molecule at position x at time t. And the way, or let me write first. Suppose you are at position n, and you can go either n plus 1 or you can go to n minus 1. And the same thing, you can from n plus 1 you can go to n, or n minus 1 you can also go to n. So there are 4 terms I wrote, because at n you can have 4 possible probability changes. One, you can from n plus 1 or n minus 1, you can jump to n. That's why you have 2 positive probabilities. And w n plus 1 n means is the rate at which, it's like probability, is the rate at which you go from n plus 1 to n. And then w n minus 1 to n, the second term, how fast you go from n minus 1 to n. So 2 positive terms and 2 negative terms here. That is, if you are at n, you can move away to n plus 1 or to n minus 1. That's why you have 2 negative terms. With me? Now this equation is a probability balance equation. That means I should. So at site n, what is the rate of change of probability? The rate of change of probability is given by 2 positive terms, which leads to increase, and 2 negative terms, which leads to flowing away from this site or decrease in probability. Okay. Now, it can happen that all probabilities, so jumping left and right, are all equal. So in that case, this equation can get simplified to suppose left and right, and it also doesn't depend on which side you are. Question? You follow? Okay. Now this looks very different from what you are familiar with. This is not ordinary differential equation, right? That's why this is called master equation. So left hand side you have like derivative, but right hand side this is discrete. You have discrete elements. So it's not, like, you cannot use techniques of ODE here. Okay. And there are various ways to get the solution. One is to use a transformed function. Okay. Are you familiar with Fourier or Laplace transform? Yes? Okay. So think this transformation is like Fourier or Laplace transform. So you cannot solve the equation in this manner, but go to a new variable by transform, like Fourier transform, and then you might be able to solve the equation. That transform is defined as GST So if you remember Fourier transform, from a function x, that's Fourier transform, correct? If you have a function of x, like gx, you can take the Fourier transform and you end up with, you can get a function in k variable, correct? By Fourier transform. So x is gone. The same idea here, that you are summing over n. So n is gone. You end up with a function in s, which is like a Fourier variable, like k in Fourier transform. So you have a new variable s. Today I'm not going to get time to show you all the steps. But let me write down 2 results, and I can come back to that next week, and you can try at home to see if you can derive those. That is, you will be able to get an equation like now this is a differential equation in time. This is a first order differential equation. And to solve this, you are going to get this is the equation. So you take this like a Fourier transform, and you change this equation, this master equation, using into a differential equation, and you can then solve it. And once you solve it, you can get back the probability distribution at 1. But what you can do, you can more easily we are interested in, we can calculate x average and x squared average, or n average and n squared average. And I can show you how you calculate n average. Your G is En e to the power INS. So dg ds is I summation n en to the power INS. That gives me 1 over I dg ds at s equals 0 is summation n en. So do you follow the steps? I had GST, this is definition. If I get DGDS, I get 1 n down. And then I take, after taking the derivative, I choose s equals 0 point. So I calculate the derivative at s equals 0. And if I put s equals 0, e to the power 0 is 1. So I end up with summation n to n, that is nothing but n average, like x average in a random one. And if I differentiate this twice and do the same steps, I am going to get n squared. So by differentiating this function, I can calculate both n average and n squared average. Okay? So why don't you try this at home, and then you can show it next month.
Hello? Yeah. I just got class. I'm going to some class, but it just ended right now. Huh? What?
Until the class break.
Oh, no, it's fine. I'm just getting my stuff right now.
I'd say those 50 degrees sounds pretty We're already done that.
That's what it says on the Oh, yeah.
That means there are 2 cells which you already set. You get hit. And then you can reverse.
You're showing like the effort. Start the so like type 2, the presentation would start after a couple hours later. Why would that?
Oh, that's by kind of stochastic fluctuation. So it's because I'm using continuous time. E is now the W's are taking the place of P's. So W's are like P's. There's no, it's basically done. So for master equation, the key of the master equation variable. So that's the problem of finding the quantity that cycles. So the master equation is defined in terms of the problem. You are not calculating the problem. Let's go calculate from the simulation part. You are calculating the multiples. You are have
I've not been but I think it's